That is very important in a way because this will be important in regulation at the level of RNA and at the level of post-transcriptional modification. Remember, I just said in the last slide, I gave you an example that a gene contains many nucleotides and there are pairs of A and T and G and C, and this sequence of the nucleotide is very specific for every gene. But give you an interesting idea, maybe you already know that all these DNA nucleotides that are present over here, they will not be playing role in protein synthesis. So interestingly, these nucleotides are all DNA nucleotides. They will be recognized by the same enzyme, but ultimately they will not be entering into the process of protein synthesis. So you have two types of sequence in a gene, in a DNA. Every gene is DNA. Every gene has introns and exons. The introns are the part of the DNA or the sequence of the nucleotide that will not be present in mature messenger RNA. So when they are not present in mature messenger RNA, they will not be involved in the process of protein synthesis. It means during the next stages of the regulation, these intronic sequence will be disappearing, or there will be some special mechanisms that will be omitting these introns. While on the other hand, the exons are those nucleotides that will be present in the mature messenger RNA or mature RNA, and they will be used in the process of protein synthesis. And if you just have a look on this colorful diagram, you will see that this is a structure of DNA structure is the structure of a gene, and there are double-stranded DNA molecules that are shown, but you can see the first part is present without a colored box, that is the intronic sequence, and then there is a blue color box which is exon, then intron and exon and intron and exon. It means nature has overlapped the DNA sequence. Some sequences are considered as intronic sequence and some sequences are considered as exonic sequence. And in the RNA level, in the next level inshallah, we will be discussing that all this DNA, all introns and exons, they are converted into RNA. But at the third level of the fourth level of regulation, when the maturation of the RNA will take place, now in the last line you can see that only the colored boxes are present. The region in between the colored boxes, they are spliced out, they are removed, and we will see how this happens. But the take-home message over here is that all the DNA is not part of the final mature messenger RNA. There are certain parts they are removed, and those parts that are not present in the final messenger RNA, they are called as intron. There are some interesting things about intron as well, that about 70% of our genome is intron. So interesting that exons are only 30%, so it means only 30% of our DNA is actually protein coding, while 70% is non-protein coding. So falsely or mistakenly, previously this region, the introns were considered as junk DNA, useless DNA. But just for a moment, think about it, how 70% of our DNA can be junk, can be useless. So now we know that these introns, although they are not present in the structure of the protein activity, they are very much important in regulation. And in the next levels, we will be again and again talking about introns, that how they are involved in the process of regulation. So you must know that introns and exons by structure, they are not different. They are all DNA nucleotides. And how they will be different, and what are their uses. Exons will be used to make protein, while intronic sequence, they will be used for the regulation of the protein, and when this protein should be formed, and whether this protein should be formed or not to be formed. So more interesting things will be coming your way, but at this moment, I just want to tell you that many genes will be exposed on chromosome. these genes are different regarding their composition, regarding AT and GC content. But what is common in all the genes is all genes are double-stranded DNA structure made up of DNA nucleotide and consist of intron-exon overlapping sequences. So we learned that only the decondensing a chromosome will not do the trick. Once you are successfully unpacking a chromosome, you will come up with lot of genes, and next level is that all those genes that have similar composition, they will be handled together. So it means those genes that have similar AT content and GC content, they will be going to the next level, and we are talking about S-phase DNA replication. So now you can imagine that when many different part of the chromosomes, they are decondensing together, hundreds and thousands of genes will be exposed, and now some of the genes present on chromosome 1, some of the genes present on chromosome 10, some of the genes present on chromosome 20 have the same composition, have the same AT-GC ratio, and they will be handled at the same time. So what will happen during the DNA replication? A gene is selected somehow, and now this gene wants to replicate, it wants to make a copy. So remember that the DNA replication is a three-step process. And obviously, when you say three-step process, you can imagine that many proteins, many enzymes are involved in the process of replication. And from where these enzymes and proteins will come? Right, obviously from the cytoplasm. So if the cytoplasm contains these enzymes and proteins, only then you can undergo the process of the replication. So in the process of replication, what will be the three steps? The first step we have briefly already discussed. The first step will be denaturation of the DNA to make the DNA unstable. Because if the two strands are attached, then how you will make a copy? First you have to denature the DNA, unstable the DNA. And the second step will be attachment of the primers. There will be specific RNA stretches that will be attached to the unstable DNA, and technically this process is called as annealing. So in the next slides I will be explaining what are the primers and what are their role. But remember, first thing is to denature, separate the two strands, and second thing is to attach a primer in this unstable DNA, and third thing is the function of the DNA polymerase, the enzyme which is responsible for replication. And DNA polymerase will be extending the primer so that one strand of the DNA will make the missing strand, and that is why we always say DNA replication is semi-conservative, that you show one strand of the DNA and the other strand can be formed. So just have a look on this handmade diagram. It is explaining the two steps of the process of replication. There you can see there are two strands of the DNA, 5' and 3' facing oppositely each other, and these two strands are separated. How we will see. Then a small part of RNA that is attached on both strands. Obviously these two strands are anti-parallel, so primers are attached on opposite directions. And then from this primer, the nucleotides will be added to this part, and then one strand of the DNA will duplicate the other side, and the other strand of the DNA will make the missing part. So in the next slides we are going to discuss these three parts that are involved in the process of replication, and we will see how the variations are also existent in the process of replication. So we have many genes that are present on a chromosome, they are exposed, and now the system wants to undergo DNA replication, and as we know, the first step is denaturation. Remember, the two strands of the DNA, they are attached through hydrogen bonds, and hydrogen bonds are considered as comparatively weak bonds. We already know that there are two types of bonds existing in the structure of the DNA. There are phosphodiester linkages that are existing within one strand, because nucleotide of one strand, they are attached with one another through two ester linkages that are mediated, synthesized, generated through the phosphate, and that is why there are phosphodiester linkages that are existing in one bond. In the middle of this diagram, there are three strands shown. In the middle of the diagram, you can see two strands with the green ladder-like structures present on them. So these green ladders, they are showing nucleotides of one strand and the nucleotide of the other strand. And obviously all these nucleotides of one strand, they are attached with one another through phosphodiester linkages. And if you look at the first diagram, which is on the left side of this slide, you can see two strands of the DNA, they are attached with one another. And we know that the two nitrogenous bases are attached through the hydrogen bond. So hydrogen bonds are comparatively weaker. If you want to break this hydrogen bond, usually you need very high temperatures. And you may already know that DNA is stable up to 80 degrees centigrade. And if the temperature increases beyond 80 degrees centigrade, usually 90 to 95 degrees centigrade, the hydrogen bond, they start to break. So in this diagram, you can see that if you will provide the heat, the two strands will separate. But keep it in your mind, the process of separation of two strands will be varying from gene to gene, and for AT-rich genes, you will need comparatively less heat. For GC-rich regions, because you have to break more 3 bonds, you will need more heat. So it means that the first step is to unstable the DNA structure, and usually it is the high temperatures that will be separating. And the question comes in our mind, from where this high temperature will come. The answer is through metabolic processes. Because in our cell, every time we have different reactions, some are exothermic, some are endothermic. So nature has very beautifully and tightly coordinated different reactions with metabolism. Because in the denaturation of the DNA, you need high temperature. So this occurs at a specific time when the exothermic reactions, heat generating reactions are occurring. So in this diagram, you can see that if you can unstable the DNA by providing the heat, you can also stabilize the DNA by lowering the temperature. So at high temperature, the two strands of the DNA will denature, and when you will cool down the cell, when you will cool down the system, the two strands will be hybridized again. You will see the more details of all this process in cell and molecular biology, but here I am just giving you the overall process and want to give you an idea that how all things are not happening at the same time. So from this slide we learned that all the genes that were present on one decondensed chromosomal part, they will not denature at the same time, because some genes need less heat, less temperature, some genes will need more temperature. So we will be talking ideally that what will happen if a gene gets denatured. Then we come to the next level, that the primer will be attaching to this unstable DNA, and then we will be talking about how the extension will take place. So by denaturation, many genes will be unstable. Obviously, if some genes have same composition, they all will be denatured at the same temperature. So at this time point, when you have provided a specific heat, there will not be only a single gene that is going to be denatured. There will be many genes present on different decondensed chromosomes. Even on the same chromosome that you are talking about, there will be many genes that will denature at the same time, because they will have the same composition, same bonding, AT and GC pairing. So now you have many genes that are denatured, many DNA that is present in a denatured form. Again, out of all these genes, nature will not undergo replication, all of them together. Again, there will be pick and choose. So The first level was all genes were not stable, unstable at the same time. You got them, many genes unstable. The next level of regulation is all of them will not undergo replication at the same time. How nature will ensure this, that all genes will not replicate at the same time? This is done by the attachment of primers. What is primer? What is the attachment of primer is for? This is basically a concept of tagging. Nature do not do things randomly. Nature always do the thing in a sequence specific manner. So when many genes are unstable, now enzyme needs some tag that which gene should be replicated at this moment, and nature uses small stretches of RNA nucleotides that are usually 15 to 30 nucleotides long. They will be binding to the DNA, and they are called primer because they will prime the process, they will initiate the process of replication. So now point comes in our mind from where these primers comes and what will be their role. So remember that primers are small stretches of RNA, and we have a concept from the central dogma that our genes produces many different types of RNA. Almost all these RNAs goes into the cytoplasm, play their role directly or indirectly in the protein synthesis. But after their specific role, they are degraded. And maybe you already know about the enzymes RNases. RNases are the RNA destructing enzymes. What they do, they break the phosphodiester linkages present in the RNA structure and break them into pieces. Remember, RNA will remain RNA until its structure is intact, but if you cut it into pieces, this is no more RNA used for the protein synthesis. But as we discussed many times, nature uses one chemical for several purposes. These degraded RNAs, the fragments of the broken RNAs, they will be used as primers. This is one of their use. And the primers are used for the process of starting the process of replication, because there are some specific enzymes that will be attaching these primers to the unstable DNA. If you go back again on the slide 6 where I had shown you the 3 stages of the replication, you will see the primers are attached, but now if you make a second careful look on that slide, you will see that the primer sequence is complementary to the DNA sequence. If there is a G sequence on the nucleotide on the DNA, this will be facing a C nucleotide of the primer. The only difference is the primers are made up of RNA nucleotide. So it means the primers can only bind to some specific unstable DNA sequence, and that is why the primer acts as a tag. Obviously, the DNA and RNA will be binded together with the help of enzymes, and we know all enzymes are not available at the same time. So it means you have thousands and thousands of RNA fragments that are floating in the nucleus and in the cytoplasm. They are act as primer, but only when they will be acting when the DNA is unstable, when some specific enzyme is available that can attach the primer to a specific sequence of the DNA nucleotide. Before listening to the next clip, you go back to slide number 6 and have a look again at what is the sequence of DNA and what is the sequence of the primer that is attaching. The rule remains the same at GC, but as RNA do not have thymine, so you will see that adenine present in the DNA is binding to the uracil of the primer, and guanosine of the DNA is binding to the cytosine of the primer, but it is a attachment between DNA and RNA. So it will be interesting to tell you something about nucleic acid interaction as well, because this also plays an important role in the regulation of the protein synthesis. But before I tell you about nucleic acid interactions, go back to slide 6 and have a look how primers are attached to the DNA.